Today we start our presentation with Dr. Jacob. As you know, that 10 years back, if you look at top 20 drugs by prescription by sales, most of them were small molecules. But today it's very, very different. The scenario is that in top 20 drugs, we have more than 12 recombinant and biologic drugs, which are so critical in saving lives. Today, Dr. Jacob is going to be talking about biologics and biosimilar challenges and opportunities. Dr. Jacob, please.
Thank you. Thank you very much, Dr. Mishra. You can hear me, right?
Yeah, sure.
Yeah. You said it very right. I think over the last decade or so, the number of big products, especially sort of the big blockbuster products in especially therapeutic areas like oncology, immunologic drugs, etc., has significantly shifted to recombinant proteins and monoclonal antibodies, and even over the last maybe 20-25 years. The manufacturing processes as well as the formulation development processes for biologics molecules have evolved and our understanding has significantly improved. Right? Unlike small molecule, drug products or APIs, the challenges associated with having a stable formulation for biologics is very different. I think a lot of complexities other than what you encounter for small molecule products exist in biologics, and a lot of progress has been made, but we still are restricted to, for almost all biologics products, storage temperature at under refrigerated conditions. Right, you still need storage at 2 to 8 degrees centigrade. There is still having a liquid formulation or even a lyophilized formulation for that matter for a biologic product, which is stable at room temperature, 25 degrees centigrade, is still sort of the holy grail for formulation development scientists in the field. Mm-hmm. Right? So that continues to be a challenge. Some of the early biologics products, they, because of lack of stability in the liquid state, used to be lyophilized formulations. Now, significantly it has shifted to liquid formulations, and I think we have identified conditions, whether it is pH, excipient, several conditions have been identified that can achieve, optimal stability in the liquid state. Mm-hmm. The other, big development that has also happened is, especially with monoclonal antibodies coming in, and for a lot of indications, especially where subcutaneous injections are required, where the volume of injection has to be small. Sometimes you need to go to very high concentrations of the protein or the monoclonal antibody, right? Sometimes it's as high as 150 milligrams per ml. Yeah. And then you run into a whole lot of additional challenges like viscosity, higher propensity to aggregate, and so on. Even those now, have been overcome over the last several years. You have commercial products available in the market at very high concentrations, which are stable for 2 or even sometimes more than 2 years at 20 degrees centigrade. So a lot of progress has happened, but still, even in fairly fundamental, there's fundamental research work happening still on predicting what could be the duration of the shelf life duration or the real-time stability at 2 to 8 degrees centigrade based on accelerated stability data. Right. And accelerated, 25 degrees centigrade or even higher temperatures. Right? So, so there's still fundamental research work happening there, and given that the degradation mechanisms that you have are multiple, right? I mean, it's usually not just one pathway for degradation that exists in such complex molecules. You have a host of different degradation pathways, and typically the thermodynamics, the Arrhenius kinetics that you use for small molecules may not be applicable for large molecules. So that still continues to be an active area of research. Coming to biosimilars, at least scientifically, the challenges are not different. I mean, at the end of the day, they are still biologics molecules, and typically the problem becomes simpler because the innovator company already has done a fair amount of work on the innovative product. and you know much more than when you have to start with a new molecule. But the challenge sometimes that you encounter for biosimilars is, patent or intellectual property issues.
Dr. Jacob, I want to ask you a quick question here. Do you think FDA is lacking in not pushing the innovator to lay out its fundamental host versus vector system, such that the glycosylation profile of the biologics, which is required for the biosimilar teams, becomes simpler and not a time-consuming and a wasteful time activity, which Amgen maintains that there's not even a single molecule in the market which is erythropoietin-like, because all the glycosylation parents are different. Do you think FDA has faltered in this?
It's a good question, Dr. Mishra. I think it's see how much control does FDA have on a subject like this versus what's the intellectual and confidential information that belongs to the company that develops a product. I think it's probably a complex area to have a straightforward answer here. I think you're right. I think a company that is developing a biosimilar has to do extensive work to figure out what's the right glycosylation profile to be met, etc. And at this point, my feeling is I think a lot of information that the innovator has is confidential information, right? And I don't think the regulatory agencies.
The structure should, sir, the structure should not be confidential.
I'm sorry?
The structure of the molecule should not be confidential, because glycosylation is part of the structure.
Right. I think so. If you look at the patents or even what may be publicly available in scientific literature, etc., whether it is coming from the innovator company itself or from other groups who may be researching on those molecules, certain like the amino acid sequence of the molecules will be known, right? I mean that's there, but the same level of information may not be available on the glycosylation profile, right? So I'm not sure again whether that information should be confidential or whether you can force companies to disclose that information. I don't have a direct answer at least to that question.
Mm-hmm. Okay. Go ahead, please go on ahead.
Okay. See another area where a lot of progress has been made, especially with respect to formulations and drug delivery, is the devices, especially when we come to subcutaneous drugs that can go the subcutaneous administration route, right? Initially we had simple pre-fill syringes, and now we have gone to fairly sophisticated pens or cartridges where you can have really accurate and very precisely controlled dosing regimens, right? Where the patients or the subjects themselves can do the administration at home. And that's an area where I think a lot of even collaborative work between pharma companies as well as companies like BD or West, a lot of progress has happened there. Right? So I think in terms of drug delivery, also we've made a lot of progress. Mm-hmm. And that still continues to be an area of progress. I think we are now talking about microneedles and even biologics molecules that can be encapsulated in microneedles, etc., which could make delivery much more easier, and from a patient compliance point of view, these may have benefits. So that still continues to be an area where there's a lot of opportunity. Even biologics, there are a few molecules, at least, insulin is a good example where people have gone from parenteral routes to even, the inhalation way. Unfortunately, insulin has not worked. I think at least the Pfizer product, was recalled, about a year or a couple of years after it was launched. There were several issues, but I think, again, I think there is still a lot of research happening in that area where whether you could, you develop dosage forms that are not parenteral, right? There's some literature around oral delivery itself, but those, I think there are really serious obstacles and challenges there. Some of the inhalation, there's some success has happened in inhalation, but again, not too much. So those still continue to be areas of research. Yeah, so I think there are several other, and I think some of the other panelists would touch upon,
Yes.
other complex.
Yes. I think this is a very, very important, Dr. Jacob. Thank you so much for discussing critical issues in biologics and biosimilar. Next speaker, we have Dr. Jayant, who's looking at, will be addressing quality issues and non-compliance in marketed drugs of RDNA origin. We have he's going to be touching on all kinds of non-compliance issues for RDNA product which the FDA faces. Dr. Jayant.
Yes, thank you for giving me this opportunity. This is Jayant. My topic of discussion today being the quality issues and the non-compliances in marketed drugs of RDNA origin. So I would like to split my talk in say about 5 to 6 small fragments. I will read it out for you: introduction and relevance. Secondly, the type of non-compliances of the quality issues which are major or most commonly observed. Then the mechanism for identification of those non-compliances. I mean, what mechanism the regulator have in place for identification of these non-compliances. Then the type of recalls. I would like to, I will shortly touch on to this topic: type of recalls and guidance for recall is issued by the CDSCO as a guideline. Some legal provisions, major legal provisions under which we can initiate actions, disciplinary actions. And thirdly, I have just added from my experiences, what are the real reasons for non-compliances in this particular, in this major area, I mean, which is of the current thrust area, that is the biosimilars, especially with reference to the biosimilars. So I would just briefly cap up with my relevance introduction. As we all know, ensuring quality, safety, and efficacy of marketed drugs is of paramount importance throughout the life cycle of the drug. The drugs of RDNA origin in modern medicine plays a very vital role in critical patient care. Because of RDNA technology, we are able to provide the patients drugs of biological origin with very high purity and quality, which was otherwise in earlier days very cumbersome and a costly exercise. And the drugs were also too expensive. Today in the country, RDNA drugs originate from 2 sources. That means what we have in India, the 2 sources imported and the indigenously manufactured drugs. Also, the biosimilars are occupying almost about 60 to 70% market share of RDNA drugs. So it is a huge thrust. With huge uplift in the indigenously developed biosimilar drugs, it becomes imperative that the manufacturers and importers must assume a responsibility and must ensure quality, safety, and efficacy of drugs of the what they import or what they produce for the Indian population. Then coming briefly to the major type of non-compliances observed. It is usually it is the failure in the assay or in the pH, which is the most commonly what we are encountering, or what feedback we get regularly. And
I want to ask you a quick question here.
Yes, please.
It is my experience.
Yes.
That managing directors of recombinant companies oftentimes give the responsibility of formulation of recombinant products to those who have been formulating small molecules. Now, if you have a situation like that, people who do not know how to handle the recombinant products are given the formulating for preclinical trials, formulating for clinical trials, formulating for the market. Of course, you will have pH problems and assay problems. Would you agree with me?
Yes, up to some extent it may be true, but unless the regulator, before granting any new drug permission, has to thoroughly has to thoroughly check all these checkpoints, like GMP inspection or giving any kind of new drug permission. It is a long process. The firm has to submit all the data first. The regulator goes through the data, then physically verifies it for the GMP compliance. And thirdly, we are recently started, all the drugs are tested at our national affiliate laboratories. Whatever, I'm not talking about very complex molecules, but there are as many as 12 to 13 identified drugs, which can be very easily tested and very conveniently tested at our affiliate laboratories. So unless ensuring those quality, we don't release those drugs in the market. So that is a kind of a barrier. Furthermore, it is the responsibility of the manufacturer or the importer post licensure stage to continuously ensure throughout the cycle. That's what I'm emphasizing. They are responsible for checking the quality of their product throughout the life cycle of the product.
And then I was trying to stress on the fact that at the pilot scale and the production scale, the people who are doing the formulation, their skill set, whether they are from biologics, most of the time they are not, and that is one major non-compliance issue, in my opinion.
Yes, maybe in few or stray cases it may happen, but unless we physically verify the premises and also the biotechnology, I mean the proposals of RDNA origin, they are checked at 2 or 3 tier level. I mean, even there are some other ministries involved, like Department of Biotechnology. They do have some control over the pre-marketing activities, or the R&D or the preclinical trial activities.
Okay.
Unless they verify their premises and their overall.
Please
Yes,
proceed.
and the second non-compliance is all we know. It may be because of the visible particles or the signs of deterioration in the containers. And the third major non-compliance is the SAE, serious adverse event, and lack of the desired therapeutic effect. And fourthly, it may be the labeling issues. That this may be arising because of various factors. I'm not going to attribute to some one kind of reason. And thirdly, the non-compliances due to non-compliance to the Indian pharmacopoeia. Perfectly fine. The imported drug, they may be passing the standards of their country of origin, but as IP or Indian pharmacopoeia has started introducing more and more molecules into their official monograph, they have to comply. So in this stage, probably they are not willing to comply to the IP, or they ask for the relaxation, and that may be a major. I mean, it may be a major hurdle for regulatory issues, regulators, or for proper, streamlining the activity. So non-compliance to the current Indian pharmacopoeia standards is also majorly, commonly observed non-compliance. Then thirdly, I would just briefly touch on to the mechanism of identification of these non-compliances. First of all, the government of India, as well as the state licensing authorities, as well as the central license approving authorities, both have this kind of approval, this kind of mechanism. The survey samples. These are the survey samples. It is a plan-wise. The officers are collecting various survey samples from various premises, say medical stores, depots, randomly. It may be anything. They have been instructed to keep a strict vigil on this. So they may be collecting the samples and bring it to office, and they will forward it to the testing laboratories. So this is one of the major mechanisms of identification of the non-compliances. Secondly, it is the port offices. It is for the, especially for the port officers are supposed to keep a control on the imported cargo, imported drugs. So they are periodically they are testing the samples of the imported cargo or the imported biological drug, along with all other small molecules also. Then any complaint received from any stakeholders. Any stakeholders is a very broad definition. It may be patient, it may be a distributor, it may be a wholesaler, or it may be even a layman, or it may be whoever is a sufferer. If he files a complaint with the central licensing authority, we have to take a note of it, and we have to act upon, or we have to investigate in these cases along with the our zonal counterparts, and we have to instruct them to investigate the issue, and eventually it goes for the testing. Then the feedback from the foreign NRS. This also is a good mechanism for controlling the quality or the compliance issues, because we are continuously getting feedbacks from the foreign NRS, or we have to study. There is a separate cell for it to study the alerts, foreign alerts. Then thirdly, one more mechanism is the, as we all know, it is the pharmacovigilance, the feedback received from the pharmacovigilance center, which handles all the SCs post licensure. So analysis of the data or any special highlight or any special signal, that is the responsibility of NRA to take note of it. And the last mechanism which I am reading out, it may be a little bit feeble, because in the in the huge workload of the routine activities, this may go a bit ignored. It is the review of the critical review of the PSUR. We are improving on it, and a critical review of periodic risk-benefit ratio report, which has been replaced recently. So these are the mechanisms of identification of these non-compliances. A few I have marked them, yeah.
I think they're very, very, very specific, very important.
Yeah.
Does any Dipti Madam, is anybody from the public interested in questioning Dr. Jayant?
I did not get your point.
I was talking to Dipti Madam, whether public want to ask any questions to you.
So as of now, there are no questions.
Okay.
I'll just check with the team once more.
Okay. All right. I think in terms of quality issues for marketed RDNA product and non-compliance issues, we have a system in place which is able to troubleshoot, and RDNA products have, in my opinion, if you see one full year of FDA recalls and FDA rejects, you find RDNA product minimally there. Minimally there meaning a lot of hard work has been done by teams, regulators, by the manufacturers, by the people who do clinical trials, etc., to making sure that the quality of the product that is reaching is robustly fine. And hence, I do not find in last 1 year of whatever I checked, last 2 years also I've checked, the complaints against recombinant products is much less compared to synthetic products. Okay, very nice, Dr. Jayant. Next, I have in the panel Dr. Kundu.
Oh, please, because I need to mug up last 2 points, and there is one more.
Okay, please, sorry, I apologize. Please go ahead. Please go ahead.
Very interesting. I mean, I was just taken out from my whatever past experiences. I will not take much of the time. As we all know, that there may be the government also may initiate a recall, and the recall may be a statutory recall or I mean, the other kind of recall being a non-statutory recall. Normally, the not of standard quality drugs, I mean, which are found not to comply with current standards, they are subjected to statutory recall.
Okay.
And normally, this is the procedure. And coming to the last, I need to highlight there is one very interesting document which is available on the public domain. That is the guidance for recall, including vaccines and recombinant. It is very explicitly said here. It is released by CDSCO, and the latest version being 2017. So if you go through that document, you will clearly get a picture what is a statutory recall and what is a non-statutory recall. What are the type 1, type 2, and type 3 type of recalls, and how the manufacturers and the importers should assume their responsibilities. It is very clear there. It's a 17-page document. Forget about all those. I mean, there are some legal forms that is not to be attended. However, the first 5 to 7 page number, they are very explicitly, I mean, mentioning all the definitions of the various recalls and the legal provisions. It has been clearly very stated that there are references of product recalls, complaints, and adverse reactions, specifically under para 27 and 28 of the Schedule M, which is our firm basis of any regulatory action or the on basis on which we can initiate our actions. So
I have a question for you regarding this quality issues in adverse reaction. This is a problem which is infesting India in a big way. I think you know that a lot of infant deaths happen due to vaccines, and these vaccines are not these deaths don't happen due to vaccine active, but mostly due to vaccine excipients in these infants. Can we not do something about this in terms of control of quality of these vaccines, such that so much of the excipient should not go into an infant?
Yeah, because that's I think it's a part of separate investigation. I mean, or in collaboration with academics, we need to do, because I don't have really any data or any experience from vaccine side. But what our regular experiences says, it is a matter of further investigation. Yes, we can initiate. Just if you allow me 5 minutes, I will highlight few reasons of quality issues with respect to biosimilars, specifically the indigenous manufactured biosimilars. Why does this quality matter arises again and again? The first most reason, what we think, is the lack of proper animal model selection. Unless usually less responsible animal models are selected by the biosimilars. just to save the time or just to, I mean, I don't know, because it may be their company policies. We don't want to go into it. They are not following, and they are not keeping in parallelity with or in line with the innovators. So it may the data may be acceptable for the regulator. However, it may pose some problem in the future. Secondly, it is usually the CMC PCT data is generated only for 3 batches. It is just it has become a magic number. However, as per the current various international guidelines and the published papers, the data of minimum 10 batches should be expected to be generated for more robust
10 continuous batches.
Yeah, 10 consecutive batches. Yeah, yes. That's what the international expectation is there. Yes. We don't want to blame anybody. However, to generate a really robust and a reliable data, the indigenous, especially the indigenous manufacturers of biosimilars, they should exercise all the similarity studies on 10 batches. Mm-hmm. Then the third major reason, it is not aligning their specifications with the innovator. While the specifications are generated, there may be different in the manufacturing process. However, this is the most commonly employed reason, and sometimes they are either relaxed or they are sometimes very stringent, but they are not completely aligned with the innovator. So they should take into these also should be taken into account. Then there is cold chain and GDP, or the lack of good distribution practices or good storage practices. It's a very commonly found in India. Then further, there may be inadequate head-to-head comparison and waiver of the data. Actually, this is the repetition of my earlier interpretation. Sometimes the manufacturer, they ask for waiver of the data or the curtailing of the data because of lack of resources or lack of time or whatever. But they are passing only the minimal. They are submitting the data of only the minimum requirement is qualifying, and they are not willing to generate more and more data and over and above the expectations to get a robustness in their data. Then poor GMP compliance post-license stage. Now this is again a matter of debate. Especially, I would like the indigenous manufacturers to be careful about complying to the GMP issues post-licensure stage. Yes, it's up to the licensure stage. They are very serious. Still, our regulators inspectors they are conducting properly. However, after as the time progresses, the grip over the GMP compliance is wanes. And the second and the last observation, what we have observed is the secondary importance to the RMP and the pharmacovigilance. RMP, risk management plan. They are submitting a risk management plan. However, they have very feeble control over it. And the pharmacovigilance activities, they are normally ignored. They should not ignore it. So that's all I want to say. Yeah.
Thank you. Very good. I have one last point on this beautiful presentation is that there are many companies who do not even have a background in rDNA because it is a lucrative business. They get into it and they have all kinds of pharmacovigilance problems.
Yeah, they don't set up an pharmacovigilance mechanism. Even they don't have team. Even they don't know there is a pharmacovigilance.
And this problem is going to grow, sir. It is not going to. This problem is growing, and you need to watch this problem on the dashboard. Yeah.
Yeah. Even in the PSURs and everything, they are just giving only what are the non-serious side effects for the adverse reaction. And they are not highlighting any major events. Yeah.
Very nice, Dr. Jayant. Thank you so much. And now we have Dr. Kundu, who is going to be talking about regulatory issues in mesenchymal stem cell therapy and plasma therapy and COVID-19 specific MABs. Dr. Kundu.
Thank you, sir. Good afternoon to everyone. Thanks for giving the opportunity. So just to set the background. So normally when we talk about any cell gene therapy or biologics approval with FDA or FDA, CDSCO or EMA. So I think the pathways have been very clear. There are elaborate pathways with elaborate checks in place. So that's why we see probably, double-digit numbers being approved in US in last decade, and early triple-digit numbers or closer to three-digit numbers being approved in Europe. But COVID was unprecedented. COVID changed this perspective of each and everyone, whether this is the manufacturers or the sponsors or the regulators, because everybody needed to pitch in to understand the aspects and impacts of COVID, how to tackle that. So the result was a lot of work was done by regulators to understand how we can fast track the emergency use authorizations or the approvals of various agents for COVID protection or COVID treatment, while at the same time taking care of the safety aspect as well. How to ensuring that this risk benefit profile always is in favor of the benefit, not in favor of the risk, and while curtailing the timeline massively. So I'll look at the aspects from regulatory aspects from FDA perspective. I think that's where the maximum amount of work has happened. Typically a very conservative organization when it comes to approving a product, especially when we say biologics, right? What FDA did for the regulatory aspect is they established this coronavirus treatment accelerated plan, and also accelerating COVID-19 therapeutic interventions and vaccines, which looked at your all aspects, whether it is your COVID-related biologics, COVID-related combinations, human stem cells, other cell therapy products, your immunomodulators, so your plasmas, hyperimmune globulins. So all of that were being assessed for emergency use authorization or for approvals in a very specific and fast track pathway. So just to give the comparison, so more than 670 development programs have been initiated for COVID in last 2 years. This is the dashboard from FDA. So I'm just quoting the numbers which are there in public domain. 470 plus trials have been reviewed by FDA. Out of that, there are 11 emergency use authorizations and 1 approval. But when we compare, say, probably 10 or 20 approvals in biologics in last 15-20 years versus 470 trial reviews in last 16 to 18 months. So that is massive. So, and out of all of these, it's not like, say, we just talking about mask and gloves and all. We talking about 50 plus antivirals, talking about 60 plus cell and gene therapy products, talking about 130 plus immunomodulators, which are being assessed, 60 plus neutralizing antibodies, and 40 plus combination of these various factors. So people are trying that, okay, let me try a neutralizing antibody with the immunomodulator, or let me try this with a IL-1 or IL-6. So all sort of combinations have been tried. So how does a regulator like a FDA or a DCGI or a EMA, they're trying to handle it? So what FDA did is, after this thing, like, say, I'm saying CTAP and active, this was established. So there was from the preclinical aspect itself, development aspect. So they started having this pre-IND meetings, pre-IND consultation programs, as well as the interact programs. So basically you have initial data, you can have a pre-IND meeting with them, you can have a single patient emergency IND application, you can file with them. For preclinical, they started helping with validated animal models, validated assays, providing access to BSL-3 labs, so that if there are 20 candidates, they do not end up spending 1 year just to find the right animal model, right validated assay, or even finding the lab over there. For clinical, typically if we see for a monoclonal antibody, the typical is like 18 to 24 months is the trial duration, then another 1 year for your immunogenicity, and then the data assessment. So it's a 3-year, 4-year program just for the complete clinical development. That's, I'm saying, on the aggressive side. Now what they try to do is to ensure EUR can be assessed while assessing the safety aspect as well. This master protocols were drafted. So master protocols were followed with a single control arm, doing the rank potential and inventory of all these molecules, all these candidates which are there for them, so that this clinical trial synergy can be executed. And how, say, India, number of patients might be higher, but in US they needed to pull in all their existing networks of clinical trials, like, say, AIDS clinical trials, or your viral clinical trials. So all of those were pulled in to ensure that this clinical trial synergy is provided to the candidates which are being assessed. Now coming to specific programs. So for biologics or biologics combinations, so couple of molecules they've been granted EUAs on the basis of the initial data, while the clinical trials are ongoing. So this is not a substitution for the approval. So this is just emergency use authorization. Once you get it, it's not that the trials can be stopped over there. Trials need to continue as more data comes in. So then they can file for the approval. As of now, only remdesivir is the only molecule antiviral which has been approved for treatment. So other molecules like casirivimab plus indemivib combination from Regeneron, whether it is baricitinib plus remdesivir combination, or your bamlanivimab and atezolizumab combination from Eli Lilly, all of them have received emergency use authorization only where the trials are ongoing. So most of these molecules are either IgG1s and a combination of NAbs or IgG1 antibodies over there, with baricitinib being a JAK inhibitor and your remdesivir being a RDRP. Now coming to plasma, convalescent plasma. So convalescent plasma, only high titer convalescent plasma has been approved for emergency use authorization. Low titer convalescent plasma is not authorized. And this plasma is a very critical subject to ensure the collection. For convalescent plasma, low titer convalescent plasma is not authorized for use. And what has to be ensured by FDA, CDSCO, or any of the regulatory bodies is the collection of plasma, screening of that, manufacturing, the right manufacturing, the complete step of the process that has to be managed before it can be used in the patients. So low titer plasma has been taken off this EUA list. Then another antibody which has been monoclonal antibody which has been given emergency use authorization is sotrovimab. It's again, I think, IgG1. Not sure about this antibody. Then immunomodulators. Probably 70-80 immunomodulators were studied, but as of now, only 3 have the emergency use authorization. Dexamethasone as a steroid. Then we have tocili, IL-6, tocili or saralimumab, and baricitinib and tofacitinib as your JAK inhibitors. No other JAK inhibitor also has a emergency use authorization. So other treatments which are mentioned, say for example, anakinra, fluvoxamine, or your inhaled steroids, or your baricitinib combinations with IL-6 or IL-1s, your canakinumab, colchicine, siltuximab, all of them, they do not have emergency use authorization. And in fact, FDA in their recommendation, they're recommending against the use of these as a for any of the moderate or severe case of corona. Now coming to stem cells, coming to cell therapy. So potential wise, if we see concept wise, it should have the largest potential. So because if we see mesenchymal stem cells, they can help you in acute lung injury. They can help you reduce the cell mediated inflammation response. And since they do not have this ACE2 inhibitors, so there should not be any chance they should be resistant to infection. But multiple trials have happened for mesenchymal stem cells. There was a trial done in China for a small number, like 10 to 15 patients. Then there were clinical trials done in US. 3 to 4 trials have been done. They tried to randomize the trials, but the trials could not be randomized in 1 or 2 instances. And then other trials also, the statistical significance number showing that mesenchymal stem cells have any significance over any of the emergency use authorization numbers. So those trials have not given any conclusive evidence. So FDA, as of now, the status of the latest status as of FDA is this is they do not have statistically significant evidence to advise for the use of mesenchymal stem cells for emergency use authorization for COVID infection.
Dr. Kundu, I want to stop you here and kindly answer this question. Do you think FDA went wrong in choosing mesenchymal stem cell to be a drug rather than a clinical protocol, because comparing a personalized medicine like stem cell therapeutics into a patient who is almost on his deathbed, getting a regulatory compliance, it's a headache for the clinician. Would you agree?
It is, see, so out of all the regulatory agencies there, so the maximum evidence generated for any stem cell, any cell therapy product, whether this is stem cell or cell therapy, has been there with FDA, whether it is preclinical models and others. So I think they've been, in my opinion, they've been right in treating it as a drug, sir. So COVID being a special situation, emergency use authorization has been given for lesser products to answer to your point. But at least I concur with this thought that any cell therapy product, whether it is stem cells, whether it is your CAR-Ts or your NKs, any of them, they should be treated from beginning till the end to the delivery of patient as proper drugs only, with the sterility, manufacturing, storage, everything to be treated at par with biologics. That's my assumption.
Okay. Thank you.
So any questions? I just wanted to keep it very small, just like an outlay of what FDA has been thinking, what has been the progress over there.
Sir, no questions from the audience.
Thank you.
Thank you, Dipti.
Great, very good, wonderful, Kundu, sir. Next is
Thank you, sir.
Dr. Bangaru. He's going to be talking on FDA recalls due to post-marketing surveillance data in clinical program.
Yeah, thank you, Dr. Mishra. And I would also like to thank the organizers for inviting me, and Deepthi for making the introduction of me. Okay, so coming to this recall procedure, I would like to dwell upon the general procedures followed in US FDA by US FDA, and also the Indian CDSCO. So basically, for the whole world, I think FDA is a torchbearer for this kind of recalls, because they very effectively follow this recall procedure and keep only the authorized products and to be safe and effective in the market. So what is a recall? Recall is generally a removal of a product for a correction, which is basically done by the regulatory agencies. And there's a small difference between recall and withdrawal. Withdrawals are generally made by the companies on their own without any legal issues, because removals can attract some legal action, but withdrawals are voluntary and they're not going to be quality impacted. So that is the difference between the recalls and withdrawals. And often we see withdrawals from the marketing authorization holders for packaging defects or some minor things. They will price changes or something, they will withdraw those products. But the recalls we can classify into three classes: class 1 and class 2 and class 3. Class 1 would be more severe, which has an impact on the quality of the product and the efficacy of the product, which need to be recalled immediately. The timeline is 24 hours. Within 24 hours, they need to evacuate those products from the market. And similarly, even in DCGI Act, as Dr. Jayant was mentioning, most of the guidelines or the procedures are imbibed from the US FDA processes. So the categorization also, the same kind of categorization also they made in DCGI also. So I have seen that 2017 document, which is used for recalling the biological products. So I think it's more or less the same. And I hope that the same thing is followed even by the EMEA and other countries. But we don't find much data from other countries. Whereas US FDA, very yearly they publish this recall history and the recall data they published every year. So class 1 recalls, as I said before, so they are very, very impacting the quality of the product, and it's directly making impact on the people's health. So they should be removed immediately. And second class of recalls are, so they are not very impacting on the health, but they're definitely they are required because the adverse events which are there from the class 2 are they are reversible kind of an adverse events. So even if the person takes the medicine of the biological product, there is no seriousness, but those adverse events can be reversed over a period of time. The third category of the compounds are, they are not going to impact on the product quality or efficacy. So they are only violative of only some of the minor things which are packaging kind of things. So they can be corrected easily. But as a statute, they have to be recalled. Any small mistake in the packaging leaflet or something, so they need to be retracted from the market. That is the class 3. So only these 3 classes are defined in our DCGA CDSCO document as type 1, type 2, type 3 kind of thing. But basically they are the same. So the biological product statute are defined in US Code Title 42, Chapter 6A, Subchapter 2, Part F, and Item 262. So these are the kind of details we can get it in this thing. And there is also CFR, Code of Federal Regulations, also defines the process for recalling of this product. So basically, there is a CFR part 1271.440, so which talks about inspection and enforcement of establishments described and orders for retention, recall, destruction, and cessation of manufacture. So these are the things defined by the US FDA. The timelines, as I said before, the class 1 kind of things, order will be effective immediately. And for class 2 and kind of things, there are timelines defined as 5 working days from the date of the authorities making this order. India also, it is like within 24 hours for the type 1 kind of thing, 24 hours to 72 hours of class 1 recalls. That is given by the CDSCO. And for the class type 2 kind of things, there is about a weekday's time given there. Recall classification of the product or batch in question shall be based on the risk assessment in line with the recall classification given by the definition. The recall notice is given by the authorities, and after receiving that, it is the responsibility of the manufacturer or his distributors to recall the product. Even the same thing is in US FDA also. So FDA doesn't go and destroy those products. So FDA, the firm has to conform with the regulations, and it has to withdraw the product. I mean, recall the product and do the necessary action, destruction or whatever that's given as that. So I would like to I have compiled last 4 years recall history in US FDA, because US FDA, as I said before, so they will be publishing the drug recalls elaborately. So in 2021, there is one class 1 recall. One is for device and one is for tissue-derived products. So total events of this class 1 are 2, and I did not find any recalls for class 2 and class 3. And 2020, there is no data available. Maybe because of the COVID emergency, probably this data must be available, but they have not compiled and presented there in the website. In 2019, this is a very, very interesting kind of a data. I looked at. In class 1, there is one recall for the device and one for the tissue. And there is class 2 for blood products. There is 289 products are class 2 recalls, and blood-derived products 3, and device 5, and source plasma 30, and tissue 13. Total coming to class 2 events are 340. So the same kind of a classification, more or less, like 2019 and 18, there is a demarcation, but more or less the same kind of a trend is seen. Class 1, again 2 events. One is, I mean, of the 2 events, both the events are from the tissue-derived products only. And class 2, blood products 305, and source plasma 21, and device 5, and tissue 15. So totaling to 346. So if you look back in 2019, it was 340, and 2018 346. Very, very close. I mean, this kind of things do happen. That means that gives an impression that so these kind of things do happen irrespective of how much efforts we are putting, but still the class 2 kind of errors are bound to happen. But only
Dr. Bangaru, I would like to interrupt here to decrease the number of recalls.
Yeah.
To decrease the number of recalls, would you think that whether it be facility, whether it be environmental problem, whether it be product or process problem, or whether it is a plant management problem, do you think the key to all this is education and training and safety awareness for the people who are running the show, running the production?
Yes, definitely. Yes, definitely.
And
because
what kind of compliance? What kind of compliance would you recommend for education, training, and safety awareness?
Yeah. If you look at this cost of the errors, the first victims are the patients, but nevertheless, even the pharma companies are also will be a victim because their product has been approved and they have all the processes in place to comply with the regulations, but there are some aberrations. Maybe it's human error or the process errors. So I mean, it's like some aberrations have occurred. That's why the recall is happening. So the pharma companies or the biological companies, they should exercise more caution and vigilance during the manufacturing and input quality. I think that's what they need to increase. So this is what my suggestion, that so the training is one thing and input quality is very, very important. So if you I mean, most of the things that tissue derived products and the blood products, so the input quality actually it's making the difference. So that is what I could see. I would like to tell one more thing, that very one case study I wanted to present. So in 2010, Amgen initiated a voluntary nationwide recall of certain lots of Epogen and Procreator, Epoetin alpha, because they have seen thin glass flakes in the product, these products, and which were found not to be seriously impacting, but definitely they are they are the lamella result from the interaction of the formulation with the glass vials. So probably there is a change in the supplier of the glass. Probably that could have resulted, but this is very big recall. So one of the most significant recalls occurred in the last decade or so. So this actually could have costed millions of dollars for the company, but at the same time, since it did not have any impact on the people health, the consumers are not affected, but the company got affected so much. And I mean, it doesn't matter for the company because they are going to come out with a new for a corrected product, an improved product probably. So this happens because the input, as I said before, so the input material, like in this case, probably there must be, if they are investigated, they must have found that the supplier of those glass wires, they should have some change in the process, or the supplier origin may be different. So this also signifies how stringent are these conditions are for the biological products, which is very, very necessary for the firms to comply with. And in this context, I would like to ask Dr. Jayant, like, for different pharmacopoeias, the different pharmacopoeial standards will be followed. And in practice, normal oral medicines, so if in case if EU PHR is not complying, they will transfer to some other non-regulated markets. Those same products they will market to in the non-regulated products. But in the biological products, I urge the government not to accept any differentiation between the product manufactured for US and product manufactured for India, marketing, etc. So whatever they are, I mean, when they are giving approvals, they should also see that whatever the product they are supplying to US, the same product they should also supply to India also. So why should there be any differentiation in the quality of the product, which is directly going into the systemic circulation, and it's of very high potent drugs. So
Dr. Bangaru, there are some few questions from the audience. One says that what is Indian FDA's viewpoint on partial PK studies done on complex injectables, which are very long? I don't believe this is true, because Indian FDA would not do anything like this, because the FDA checklist clearly tells that PK studies have to be completed if it is whether it be a bioequivalence study or whether it would be a complete IND study. This PK study it cannot be partial, and that I believe I answer that question on behalf of Dr. Bangaru. Okay, the next question has been you agree with me, sir?
Yeah, correct. The same standards are being followed.
Yes, correct. Absolutely. There is no change in standards. It says another person says, what are the challenges biologic formulations which are different than small molecules? Very different. We see similar issues like pH issues, precipitation, potency loss. Yes, the PKPD studies in biologics is very different than PKPD study in small molecules. So these are different issues. Okay. So next, Dharmaraj More, he's asking, is there any provision to monitor submitted data statistically in authorities? Because nowadays many regulators focus towards statistical similarity or comparability with respect to complex products. The comparability issue is always, if you are a bioequivalence guy, I don't think there is anything else other than bioequivalence which can compare two complex products. Okay, so I hope Dr. Bangaru also agrees.
No, actually, it is more stringent from the bioequivalence point of view for complex injectables than the oral products, because complex injectables have multiple criteria being applied. So not only the normal PK parameters, so there will be more than those partial areas and all, they will be applied for complex injectables, because they are potent, and they are directly going into the circulation. So enough assurity of the product quality is very much essential. So that's why there will be more stringent laws than the normal oral products.
Okay, there is a question coming to Dr. Kundu. Rajesh or Sapna is asking, is there any stem cell therapy found available for cytokinesis? It's a genetic disorder.
I don't have idea, sir.
I don't think so either. I don't think it is there yet because it is right now. Stem cell therapy is for cardiac problems or neuro problems or stuff like that. But
ortho, it is being used, ortho, cardiac, and neuro problems. It's still in early stage right now.
Very early. Yeah. I totally agree with you. There is one more person is saying that, sir, it's a pity to our Indian regulator and ISO standards that is still we don't have place for our COVID vaccine made by Bharat Biotech. No, I think these are decisions taken by IMA in connection with the government, and I think we have to at best align. We believe good science is going on. We believe the vaccines have come to the Indian populace. Most of the people have been doubly vaccinated, and they're waiting for a booster. Maybe people are talking about the booster. So I believe our Indian mass is totally aligned with government and IMA. This is a classic example of how we have protected most of our people. There is deaths every day. About 200-300 people are going, but for vaccination is going on strong. And the way the vaccination has happened using mobile phone apps, using BMC coming to your doorstep. BMC came to my doorstep. Okay, so I have trust in the system because BMC came twice with just a single call. Yeah, please.
Sir, I think this is probably the best case study where this Indian vaccines for COVID, in my opinion, the best case study of last many decades where manufacturers, regulators, government, on-ground personnel, everybody has come and pulled together at the fastest, best pace available to provide the global quality, providing the safety aspect as well as access to the patient. So in this, this is my opinion that probably the best handled development and the commercialization program in at least India's history.
Exactly. I agree with that.
Yeah. Okay, there's another question. Impurities and outer specification results, ongoing stability programs for RDN products, very common. Absolutely is very common. I mean, everybody knows that. See, stability issues in RDN products, if it is there, it is handled right there with the manufacturer itself. It does not go to...
Dr. Shrikant, actually I was answering to the earlier question.
Okay.
It's not a question.
Okay. Very good. Great. I think we have had a wonderful session today with Dr. Jacob talking about biologics and biosimilar challenges, his drug delivery system, the oral focus on oral delivery, focus on formulation, focus on the structure of the molecule. Then Dr. Jayant talking about the quality issues and non-compliance issues, typically failed assays, pH test of failure, therapeutic effects of impacts, labeling issues, IP, Indian pharmacopoeial issues, import issues, animal model issues, innovator issues. These are a very important contribution coming from Dr. Jayant. Then Dr. Kundu talked about this clinical development and fast track products, how the regulators have done a fantastic job in last 2 years landing the number of products that they have landed, right? And then Dr. Bangaru talked about the recall procedure for FDA, which was an outstanding presentation from Dr. Bangaru.